Hello and welcome to our PowerPoint here for Unit 4 Area Study 1 on innate immunity. In this PowerPoint we're going to cover our first two key knowledge points. So those two key knowledge points from the study design you can see here, that's the physical, chemical and microbiota barriers as preventative mechanisms of pathogenic infection in animals and plants. And this is essentially going to be our first line of defense in the immune system. Then we also have the innate immune response, including the steps in an inflammatory response and the characteristics and roles of macrophages, neutrophils, dendritic cells, eosinophils, natural killer cells, mast cells, complement proteins and interferons. So there's a big list here of lots of different things that are involved in the innate immune response that we're going to have to go through, and we're going to learn what it is they do and what they're like. So just to give you a bit of an overview, we are doing the immune system. And it's really quite a complex system. We've got all these different cells involved. We've got lots of proteins. They help defend the body against infection. And it's really great in how we're able to protect ourselves from disease. And we can break our immune system down into what we would call innate immunity and adaptive immunity. And innate immunity is immunity you're born with, things your body can do from the get-go. Adaptive immunity is something that's acquired over time as your body adapts to what it's been exposed to. And we can break things down a little bit further here. We can talk about innate immunity being non-specific defenses, and in that you have the first line of defense and the second line of defense. For our adaptive immunity, this is where things are getting specific. It's where we're creating a memory of everything we've encountered and how we've defended against it previously, so we can mount a better response in the future. And this comes down to our third line of defense. And so you can see a bit of a summary of each one there. Our first line of defense is all about preventing pathogens getting entry into the body. It's non-specific, it doesn't care what the pathogen is, it treats them all the same way. And this includes physical, chemical and microbial barriers. Our second line of defense is what's in play once the first line of defense gets breached. It's still non-specific, so it doesn't care what the pathogen is, it just responds because it's not meant to be there. But the second line of defense is now using cells and proteins to help defend the body, and not just barriers in place. The second line of defense happens really quite quickly. It doesn't really last though once the infection's gone. So once the infection has been dealt with, the second line of defense's job is over, and so it dissipates after that. And then we get to our third line of defense. This time around, we are being specific. Our third line of defense knows what it's fighting, and it's being really specific to those pathogens. It does mean that this takes a little bit longer to get started, and so it's a slower response. It's just not able to happen as quickly because it is being specific. What we do have though from the third line of defense is it hangs around, and we have remnants of that line of defense lasting well after our infection has cleared. And that's actually really helpful because that's how we become immune or get immunity to certain pathogens. It's the reason if you have chickenpox as a child that when you're older, you shouldn't be getting chickenpox again, and that's because your third line of defense still remembers when you got chickenpox as a kid and is able to defend you against it. One way of thinking about the lines of defense is you can sort of think about the first line of defense like in war. You might have physical barriers like barbed wire, and it's not specific. Everyone gets tripped up in barbed wire the exact same way, and so that's our first line of defense. Our second line of defense is like our frontline soldiers, the infantry. They're rushing to where they need to go, but they just know that the enemy's over there and they're going to rush and they're going to fight that enemy. They don't really care who it is. They don't have overly specific techniques. They're just getting in there to get the job done and to try and quash whatever's happening. The third line of defense is more like your intelligence agencies or your special forces, where they actually know what they're fighting, they have special things that help them recognize what's going on, and they're able to target things really specifically. It might take them a little bit longer to get together to get to where they're needing to go, but they're able to be much more effective because they know exactly what they're fighting. Now, before we go much further, one thing I really want to emphasize here is that often when we talk about the immune system, and the notes that we've got here as well do this, we break things down to help put it into more bite-sized chunks to make it a little bit easier to understand. But in reality, the immune system is a very complex system that works all together. And so we talk about the first, second and third lines of defense and sort of split these up into different sections or use different terms to describe them, and it sounds like we're separating them. But in reality, the different sections of your immune system are always working together. they work together to mount a really effective response. It's not one thing happens and then the next if the other one gets breached. They're always working together, and they share a lot between them. And this is particularly true when we look at the second and third lines of defense. The third line of defense, the main reason it knows what to target is because it has messengers coming from the second line of defense who are able to tell it, hey, this is the pathogen, this is what you need to deal with. The other thing to consider is it isn't a case of the first line of defense stops and then the second line of defense starts, second line of defense stops, and then the third line can start. They're actually overlapping much more in their actions than that. And so the second line of defense may be getting going and happening in, full action, but in the background, the third line of defense is getting ready to start. It just takes a little bit longer to get going. And so we have this much more overlap between these different sections of our immune systems than we normally seem to think of it. But we do break it down just to make it a little bit easier to grasp what we're talking about in each section. But please remember that everything here is working together as one. So let's talk about the innate immune system. It's non-specific. It doesn't care what a pathogen is. It doesn't care if it's a virus or bacteria. It just knows it's not meant to be there. So the innate immune system is going to act against all pathogens in exactly the same way. It's really this one-size-fits-all. It doesn't matter who the pathogen is. The innate immune system is always active, so this is part of why it can mount such a quick response. The third line of defense, your adaptive immune system, is a bit more in the background, and it really only gets active once it's called into action, and that's when you have an infection present. The innate immune system, as I mentioned before, its defense is not long-lasting, so once it's done its job, it goes. It doesn't create a memory of previous infections, so it treats every encounter with a pathogen like the first time it's ever seen it. We do break our innate immunity, like I mentioned before, down to the first and second line of defense, and you can think of the first line of defense more like the barriers, and the second line of defense, the generalized cell and chemical responses that happen. So our first line of defense, it's about preventing pathogens from getting into tissues. And this is true for not just us, but for all animals and also for plants as well. Generally, we put our first line of defense into three categories. We have our physical barriers, we have chemical barriers, and then we have biological, microbiota, or microbiological barriers. And that last group is exactly the same thing. Small living organisms that create a barrier to other pathogens. Exactly what we do versus what plants do differs, but generally you can still group all the defenses into one of these three categories. It is worth noting plants do not have a circulatory system, which means they don't have cells like phagocytes, and so each plant cell actually has to be able to defend itself. It's part of why plant cells have to have their own cell walls. It's that extra layer of individual defense, because they don't have the ability to transmit immune cells around the body of the plant. So let's look at physical barriers. And here you can see it's all about preventing the entry of a pathogen. It's more like a wall. Sometimes this includes physical motions that also help to prevent the actions of a pathogen gaining entry. So some of the things we see in animals, first of all, intact skin. This is the outer layer of your skin is actually dead cells, and the pathogens can't infect them. So they're a really great barrier on the surface there. And plants have a similar thing. For plants, the equivalent to them would be having intact bark. And it means, again, their cells are protected, pathogens can't gain entry. Animals also have what we call expulsion reflexes. These are actions or motions that help get rid of something from within or on the body. So coughing for clearing out sort of lower down in your respiratory system, sneezing for clearing out higher up the respiratory system, like within your nose, vomiting for when something's in your stomach and it should not be there, and even itching and scratching can be part of this. So that's something plants aren't going to do because they can't move in that way. We also have in animals what we call ciliated mucous membranes. These are membranes of cells in areas where you have an opening to the inside of the body. So things like the airways. And it's a lining of cells in the airways that these cells produce sticky mucus that will help, first of all, trap pathogens. But as you can see in the diagram there, the cells also have these fine little hairs called cilia coming off them. And these cilia wiggle along or they wave along, and they can help move particles through them, a bit like a sweeping motion. And so this is something where you might have the mucus can trap the bacteria or it can trap other pathogens, and then the cilia can help sweep it up the lining of the airways to a point where you could cough it out, for instance. And that's another way that we can prevent things from gaining entry into our bodies. For plants, a couple of other things that they have include waxy cuticles. For instance, you can see in the leaf in the diagram, or in the photo I should say, that it has a very shiny surface. This is because it has a thick waxy cuticle there. Different plants, the thickness of that cuticle will vary, but it's certainly something that plants have that they can use to defend themselves, because it prevents entry into the cells because it acts as a barrier. Also, as I mentioned before, individual cells have their own cell walls to help defend the individual cells. Moving on to chemical barriers. Chemical barriers are where we're using chemicals to help stop things from growing or pathogens from getting in, or even to destroy the pathogens. In animals, a couple of the ones we have are, first of all, the acid in our stomach. Not only can we vomit things out if it gets into our stomach, but the acid in our stomach is a really low pH that actually helps to destroy a lot of pathogens. So even if they do get into our stomach and we don't vomit them out, chances are they've been destroyed by being in the acid of our stomach anyway. Another thing we have that you may not know is in your tears you have enzymes that get secreted. These enzymes are called lysozymes. Their job is to rupture and destroy things. That's where the lyso part comes in. It means to rupture something. And lysozymes are enzymes that help digest, which is a fancy science way of saying to break down something down, that digest the proteins and bacterial walls. You also find these enzymes in saliva. And the other thing with tears is there's a bit of a mechanical motion here, in the sense of the water can help wash away pathogens too. In plants, they don't have stomach acid and they don't have lysozymes, but they may have antibiotics. And so these might be certain enzymes like protease enzymes that if a organism eats it or is exposed to it, can actually help to disrupt the digestive functions of the thing that's consuming it. Eventually those organisms go, that's not a good thing to eat, and they steer clear of it, and that helps to protect the plant. You also find plants are able to be infected by fungi, and so they might produce things like cellulases and chitinases or chitinases, that these are enzymes and their job is to digest the components of fungal cell walls. And so a fungal infection may start out, but you'll find that the presence of these enzymes means that the fungus starts to be broken down and starts to die in a way that the plant can protect itself. Plants also are able to produce other chemicals. These might be chemicals that mimic insect developmental hormones, so end up stopping insects from maybe being able to procreate and therefore reduces the population of insects in the area, or they might be toxic as well. Plenty of plants certainly can create toxic chemicals, and there are definitely plants as humans we have to stay away from because they could be toxic to us. But again, these are all just ways plants can defend themselves. And you can see in our diagram there's a few things, some of them are physical and some of them are other chemical barriers there. Finally, our last barrier. This is really one that's more specific to animals, and this is where we have microorganisms helping us out. And so one of the most common ways we would think about this is in terms of our gut or on our skin. We have normal or natural bacterial flora. These are bacteria who are good bacteria for us. They live on us or in us, and they actually help us to engage with the world without becoming sick, or they might even help us with digesting food if they live in the gut. One of the big ways they help us and protect us from foreign bacteria or from pathogens is because they need the space on our skin or in our gut or wherever it is, and they are going to compete for that space. And they are going to try and compete with anything that tries to rock up that's not meant to be there. And generally speaking, they're able to get quite a good foothold on our skin or inside our gut, and they're actually able to prevent other bacteria from establishing an infection in those spaces. If something happens and maybe the bacteria in your gut get killed off, unfortunately that is an opening for bad bacteria to get in and to maybe create a bacterial infection in the gut or something like that, or even on your skin. So microbiota barriers act in the way that they are in the space. They are outcompeting any pathogenic organisms that are trying to colonize those spaces. So here's a little bit of a summary of our first line of defense. It's all about the physical, chemical, and biological barriers that our body has to prevent pathogens from getting into tissues. And so you do have to remember in some respects that we are kind of a tube. We have a hole at one end and a hole at the other, and we have a few other ways of getting inside our bodies. So we need to make sure we have all those ways being protected. So it's not just about having skin to stop things from getting in under the skin into the tissue there. It's also looking at the other entry points within the body. Those are generally places you're going to find some sort of barrier, be it a physical one, a chemical one, or even a biological barrier. So that is our first line of defense.
You may not know it, but your body is engaged in a never-ending battle. You are literally covered in staph and strep and E. coli and all sorts of dubious characters that are intent on using you and your body's many resources to feed themselves, find shelter, and reproduce as much as they want. And hey, we all gotta make a living, but it is not your job to give these guys a free lunch. So your body has developed a 3-part policy toward these shady customers, and its enforcement is handled by your immune system. The immune system is different from all the other systems we've talked about this year in that it's not a specific tissue, organ, system kind of system. Instead, it involves a whole bunch of different tissue groups, organ systems, and specialized but widely distributed defense cells. Together, this league of extraordinary substances joins forces to perform all of the defense functions your body depends on to keep you alive in an incredibly germy world. And the first line of defense in this never-ending battle, that's your innate or non-specific defense system. Like your average frontline soldier, it's prepared to immediately engage with anyone suspicious, and it mostly includes stuff we were born with, like the external barricades of your skin and mucous membranes, and internal defenses like phagocytes, antimicrobial proteins, and other attack cells. But some enemies must be fought with special forces, and here your body can deploy your adaptive or specific defense system, which is more like your SEAL Team 6. It takes more time to call in, but it's specially designed to go after specific targets, and it keeps files on those bad guys so it knows how to handle them next time around. But today we're going to focus on your innate system and look at how it uses an arsenal of physical and chemical barriers, killer cells, and even fever to keep you healthy, proving that sometimes the symptoms we associate with illness are actually the signs that we're healing. Just because something is simple doesn't mean that it can't be elegant. I mean, your body is capable of some incredibly sophisticated things, including defending itself from infection. But occasionally, there's something to be said for brute force. And a lot of your innate immune system's functions aren't exactly subtle. For example, your body's very first line of defense is a simple physical barrier. And it works. Like a wall around a fortress, your skin does a fantastic job of keeping out all manner of malevolent microorganisms. As long as that tough, keratinized epithelial membrane doesn't get torn open or busted up too much, you could probably, make snowballs out of raw sewage and still be alright. Although, no. Your many mucous membranes also provide a handy physical barrier. You'll remember that they line any cavity that opens up into the germy outside world, including the respiratory, digestive, urinary, and reproductive tracts. Not only do your skin and mucosa supply simple physical protection, they also pack some serious chemical weaponry. Eat some questionable leftovers for lunch? Don't worry, your stomach is literally filled with acid, so you probably are covered. Walk face first into your coworker's nasty sneeze cloud? No worries, your nasal passages can whip up a tissue box worth of sticky mucus to help trap viruses before they enter your lungs. You've also got bacteria-fighting enzymes in your saliva and lacrimal eye fluid, and peptides called defensins in your skin and membranes that help keep bacteria and fungi from setting up shop around inflamed or scraped skin.
Alright, so here we have a few exam questions for you. I'll read through them and then you can pause the video, go and answer the questions yourself and check the answers with the video linked in the description. So question 1 here says, the first line of defense against pathogens includes the A. Activation of helper T cells, B. Presence of acid in the stomach, C. Release of histamine from mast cells, or D. Production of interferon from virus-infected cells. Question 2 says, one of the similarities between the defense mechanisms of a plant and an animal include the A. Production of memory cells, B. Release of immune cells through a circulatory system, C. Use of an epidermal layer to inhibit the invasion of pathogens, or D. Production of salicylic acid to warn cells of an invading pathogen. Question 3 then says, which one of the following is an example of a plant defense against the pathogen? Is it A. Production of antibodies, B. An active immune system, C. Extensive cell death throughout the plant, or D. Waxy leaf surfaces acting as physical barriers? So like I said, you can pause the video now, work out your answers, write something down, and then go and check your answers against the ones I go through in the video linked in the description below. Alright, so here we have exam question 4. Not a multiple choice question this time, so chance for you to write some things down and pull that information out of your brain. The question says yellow fever is caused by a virus transmitted through the bite of a particular species of mosquito. An unvaccinated person travelled to an area where yellow fever virus existed and became exposed to the virus. Describe 2 ways in which the first line of defense of their body would protect against an infection by this virus. This is a 2-mark question, so we probably want to have 2 points here. Anyway, you can pause the video, write down your answer, and then go check it against the video linked in the description below. Alright, so now we're talking about the second line of defense. This is where we are moving from having barriers to actually have actors coming into play and doing things. And these actors are specifically cells and different molecules. And so really, if we manage to get past those barriers, this is where the second line defense is really coming into play and being activated. It's only really going to have action at the site of infection. It's not a systemic thing, it is a localized response. It is also only going to react against microbes. It doesn't react against toxins and other foreign materials. One thing to establish first of all is, how does our second line of defense know there's even an infection there? How does it know it's not us? Both the second and third lines of defense have ways of identifying what we'd call self from non-self. The third line of defense is a lot more specific in how it can do this, and this is part of why we call it a specific response, is because the third line of defense can actually identify individuals of pathogens, whereas the second line of defense just needs to know if that is you or not you. So this is what we call self versus non-self recognition. And the immune system can do this through the presence of certain types of molecules. The second line of defense is very broad, and they're just looking to see, do they have something on them that is not me? And so on your cells in the second line of defense, they have these structures called pattern recognition receptors, or PRRs, and you can see that down there, they're the green receptors sticking off the cell. What these PRRs are looking for is they're looking for molecules or structures of molecules that aren't made by you. Specifically, we call them pathogen-associated molecular patterns. So they're patterns of molecules that are associated with pathogens is what that means. The acronym for this is PAMPs. So the PRRs on your cells are looking for the PAMPs on the pathogens, these molecular patterns that are associated with non-self organisms. And so some examples of PAMPs might be in bacterial cells. They have something called a lipopolysaccharide, which is a type of sugar and fat molecule that's not something your cells make. So if that is present and it's picked up by the PRRs, then those cells know, hey, this is not meant to be here. This is non-self, and that's a problem. Another example would be double-stranded RNA, that some viruses have this. That would be an example of something those cells could pick up and be like, hey, again, not meant to be here. This is not made by me. So this is a non-self thing that I need to deal with. And so that's a very basic way that the second line of defense can recognize self from non-self. It's like, hey, this is a molecule I don't make, so we've got to attack this. The third line of defense is, like I said before, a lot more specific in what it's actually doing. It uses different types of receptors in those cells, and they're actually going, hey, I recognize this specific antigen, this is not a me thing, and I'm going to remember this for later. And that's something that when we get to the third line of defense, we'll actually dive into in more detail. Also, as we're getting to talking about the second line of defense, I'm going to bring up these 2 terms, cellular versus humoral. You'll notice that we often talk about cellular responses or humoral responses. Unsurprisingly, a cellular response is one where cells are doing the action. A humoral response, though, is a little bit more confusing. And this is actually due to using the word humour, so that we might have heard in a historical, almost medieval sense, like the 4 humours. And here we just mean fluid in the body. And so a humoral response is where molecules in the fluids of the body, those molecules are the ones doing the actions. So cellular response is a response where the cells are doing the action, and a humoral response is where molecules that you find in the fluids of the body are actually the ones doing the response, not cells. It's more applied to different parts of the third line of defense, but we do see it used in the second line of defense, just in a little bit more limited way. So that's why we're talking about it now. Now, for our second line of defense, we have a number of cells and molecules that we are going to go through. There's a whole range of different cells involved here. We're not going to go through all of them. What we are looking at are macrophages, neutrophils, dendritic cells, eosinophils, natural killer cells, mast cells, and then our two molecules are complement proteins and interferons. Now, I also have a little FYI box in the middle there, and that's about leukocytes. You may see the term leukocyte pop up places, and this is just a very general term for a white blood cell. So your red blood cells are erythrocytes, your white blood cells are leukocytes. All of the cells I've mentioned here just before, those all come under category of leukocytes. They are just white blood cells. We can get more specific in how we classify them, but leukocyte is just a white blood cell. And you'll notice there might be a couple of different spellings there because there is a couple of different acceptable ways to spell that word as well. Another type of cell, or way we might refer to a cell, is as a lymphocyte. Now, lymphocytes we tend to talk about much more associated with the third line of defense, but there was one cell on that list that technically is a lymphocyte. They're again a category of white blood cell, or they're a category of leukocyte. And really, these are cells, they're white blood cells that come from a lymphoid progenitor. There is a cell in the lymphatic system that gives rise to these. Most of the other cells we're going to talk about come from a different starting cell called a myeloid progenitor, but we'll get to those in a little bit. So the one lymphocyte we're going to look at here is the natural killer cell. Not to be confused with, you might be able to see in the diagram there under T cell, there is an NKT cell. That is a natural killer T cell. That is a different cell from the one we are looking at. We are looking at natural killer cells. They're non-specific lymphocytes. This is where they're a bit different to B and T cells. B and T cells, which are in the third line of defense, they're specific cells. They're able to recognize different antigens specifically. But because of that, they're in the third line of defense. They are also lymphatic. They're also lymphocytes that we see circulating in the lymphatic system. And so they're a bit different. But we want to talk about natural killer cells because they're actually pretty cool. And they are able to kill, or rather convince, other cells that may be infected or cancerous to go and kill themselves. And that's part of how natural killer cells work in the second line of defense. Specifically, these guys are what we call large granular lymphocytes. So we talked about their lymphocytes because of where their origins are, and they're quite big cells, hence the large, and they're granular because within them they've got these granules, or these little clumps of molecules that are all grouped together that they can get released together. Natural killer cells we would call cytotoxic. Cyto means like relating to a cell, and toxic means, well, toxic. So cytotoxic things are things that can kill cells. And so natural killer cells, because they can kill other cells, are considered cytotoxic. They're able to give this non-specific natural protection against pathogens because, remember, these are guys, they're not going around going, I know what that antigen is. They're going, hey, you're not me, I'm going to kill you. Which sounds quite harsh, but it's essentially what they're doing. You're not from this body, you need to go, so you're going to die. They're actually able to recognize when your cells have become cancerous or even infected by viruses, so they can actually help deal with those sorts of things really early on. And they're able to do that because molecules on the surface of your cells actually change if they become cancerous or infected with viruses. And because of that, they start registering as not self or non-self cells, which the natural killer cells can then recognize. And they're then able to put into action how they would deal with pathogens for these cells as well and kill them off. So how are they actually working? Natural killer cells have two receptors on them, kind of like two switches. They have an activating and an inhibiting receptor. The activating receptor is like the kill receptor or the kill switch, and the inhibiting one is the don't kill receptor or don't kill switch. And so normally what happens as a natural killer cell goes around, when it bumps into your cells that are perfectly happy, those cells are able to latch onto the don't kill receptor. And so because of that, the natural killer cell goes, ah, cool, this is a me cell, I'm not going to kill it, it's fine. However, the structure that does that attaching is called an MHC marker. And MHC stands for major histocompatibility complex, something you're going to need to remember, and something we talk about more in the third line of defense. That marker will change if the cell becomes cancerous or infected by a virus. And because of that, if it's a virus-infected cell, for instance, the MHC marker becomes affected and modified and no longer able to bind to that inhibitory receptor. So because it can't attach to the don't kill receptor, the kill receptor can now be attached with a different structure. And so what this means is the infected cell isn't able to protect itself and avoid being destroyed by attaching to that don't kill receptor. Instead, it actually attaches to the kill receptor, and it triggers the natural killer cell to attack it and to mark it for elimination. And so that is actually what the natural killer cell is doing. So it's going, are you a me cell? That's fine. Yes, that your MHC1 has been able to attach to my don't kill. It's all good. Or that cell's infected, and instead of being able to do that, the MHC marker is occupied and unable to attach to the don't kill, and instead a different molecule attaches to the kill receptor, thus marking the cell for elimination by the natural killer cell. What natural killer cells do is really smart and actually quite cool. You see, natural killer cells don't come along and cause the target cell to explode or anything like that, although that would be kind of fun. What they actually do is they're able to trigger apoptosis in the cell they're trying to destroy. They're able to get the cell to kill itself. And they do this using two really specific sets of molecules. These molecules are stored in those granules in the cytoplasm of the cell. So what it first has to do is it has to release those granules, and we call that degranulation. And what happens in degranulation, those granules get released, as you can see in the diagram on the side there, and one of those sets of granules contain granzymes and the other one contains perforin. And the perforin is the one that needs to go first, because perforin is called that because it can essentially perforate the membrane. If you perforate something, you put holes in it. And so here, perforin is a set of molecules or set of enzymes that are able to punch lots of little holes in the surface of the cell. That putting in of the holes on the surface doesn't actually destroy the cell, but it does create a way for the granzymes to get in. And those granzymes are enzymes which are protease enzymes, and what they can do is they can now get into the cell and they can actually trigger apoptosis. And so that is how the natural killer cell works. It doesn't come along and make a big show out of killing the cell. It just quietly punches some holes in the surface, inserts some granzymes, and those granzymes are able to trigger apoptosis in the cell. It may be a little bit slower, but it is a very effective process, and so it can take a few hours to fully deal with the infected cell. But that's what's happening with natural killer cells. And you might be thinking, why doesn't it just cause it to explode? It's way more exciting. Well, there is a problem there. If the natural killer cell just came along and caused the infected cell to rupture, the stuff in the infected cell can now get out and could possibly move on to infect other cells, so it actually creates a bigger issue. However, if the infected cell destroys itself via apoptosis, it actually forms lots of little contained blebs at the end of that, where you have all the contents of that infected cell contained within membrane, like blobs of membrane. And so because of that, you don't have all the things that cause the infection being able to leave and go infect other cells. They're all contained within these blobs of membrane. What can then happen is a cell like a macrophage can come along, and a macrophage's job is to come along and essentially clean stuff up by eating it. And that cell can come along and it can eat up these remaining blebs of membrane that you've got left over from apoptosis. And so it's able to then destroy whatever is in there. So whatever was infecting the cell that needed to be killed off, that doesn't get to escape and go everywhere. Rather, it's able to be contained and then it's able to be cleared up. And so you're not going to have this result of continued infection. So it's actually a really effective, really sensible way of dealing with the issue. So that's what natural killer cells, but now we're going to come to our other whole group of innate immune cells, and these come from a different starting point. So they're not considered lymphocytes. Most of these we would actually consider granulocytes because, like natural killer cells, they've got granules in them. And so this is quite a defining characteristic of them. And those granules will vary from cell to cell, resulting in slightly different effects. The four granulocytes are mast cells, basophils, eosinophils, and neutrophils. Now, you actually don't need to worry about basophils. I've just mentioned it here because it's one of the examples, but it's not one we're going to go through because it's not actually covered in the study design. So let's talk about eosinophils first. You can see some images of them down there. They are a granular white blood cell. It circulates through the body. It's always in the blood. These cells are what we would call phagocytic, and that means that they are able to eat up and consume broken down cells, bits of garbage, bits of pathogens that aren't meant to be there. And particularly eosinophils, we tend to notice that they are used in dealing with parasites. And so they're able to help break down parasites by releasing toxic chemicals that cause that parasite generally to die, and then they're able to help to break that down and to eat it up. And so these guys normally work in scenarios where you can't just come along and consume the pathogen in one big gulp as a phagocyte. You need to first destroy it and kill it and then break it down to smaller bits. We do notice that these seem to be involved in a few other places, which is not so great, and that includes hypersensitivity and inflammatory reactions like asthma. So occasionally they might get a bit worked up and go overboard in doing their job, and unfortunately that can mean that we see them in these not so great scenarios where hypersensitivity is like an allergic reaction, or inflammation scenarios like asthma. So those are a couple of other places we see them act. Mast cells don't circulate in the blood. You actually find them in tissues, and we generally find them in areas where entry to the outside is able to be gained. And so these are more like border guards. They're at places maybe at openings, like within the airways or within the esophagus and that sort of thing. And again, these are granular cells, so they have granules in them that have all these different molecules, specifically different types of cytokines. Histamine is a really important one that they have, and they also have heparin. And histamine and heparin have a couple of interesting actions. Histamine is particularly important when it comes to talking about the inflammatory response. It's also really important for talking about allergic responses and that sort of thing. And you might think histamine sounds a little bit familiar, and that's because often we take antihistamines, which are drugs that help combat what histamine is doing, particularly for things like hay fever, where that is caused in a big way by the release of histamine thanks to the presence of pollen, because that's what the mast cells are doing. They detect the pollen and they release the histamine in response to that. And so for hay fever, you'll take antihistamine to help combat the mast cells actually doing that. What histamine actually does is it's allowing the blood vessels to become more permeable. So that means stuff that's in the blood vessel is able to migrate through the blood vessel wall out into the surrounding tissue. We also notice with histamine that the smooth muscles are able to contract, and that's something it helps with. You can actually, if you think about increased permeability of blood vessels, if stuff can move out into the surrounding tissues, it's probably going to cause the swelling of that surrounding tissue, which is why we often notice swelling in allergic reactions. Heparin is a molecule that's actually an anticoagulant, and so it stops the blood from clotting, and that can be really useful at certain times, and it's actually a key part of the inflammatory response, and helps with the production of hormones involved in inflammation. So those are two molecules that mast cells will release, and they have really important responses for both of them in the inflammatory response. And again, when a cell like this releases granules, it is called degranulation. Mast cells work, like I said, they sit at the entry points generally to the body, and they're acting like border guards. And they're really helpful for that early recognition of pathogens. And so they can help recognize and detect a pathogen very, very early on, and they'll release some of these molecules to help signal to other cells in the immune system, hey, come over here, I need help, we've got a pathogen coming in, there's an infection here. So they're basically raising the alarm. As I've been mentioning, they do play a response in allergies, and it's a bit unfortunate. That's when they sort of get a bit too enthusiastic about their job, and instead of it being a pathogen, maybe you ate a peanut and the mast cell is freaking out over that instead. And so mast cells do actually play a very key role in what we would call the acute inflammatory response, and in also allergic reactions or allergies and that sort of thing. Our neutrophils are up next. There are heaps of neutrophils in your bloodstream. They are the most abundant white blood cell that circulates, and they're really your first responders. So whilst the mast cell might sometimes be the first one to pick up on something if it comes past the mast cell, they don't move anywhere. Neutrophils are always circulating, and so if something has gotten into the body and manage to circumvent those mast cells and essentially get around the border guards, then the neutrophils are going to be the first ones into the fray. They are there from the get-go. And so cells in the area for where that infection might be might release some chemical signals. Neutrophils are a little bit like a paramedic in a sense, or a police officer. They're a first responder. They get there really early on. Again, another type of phagocyte, and they also have granules, so they can degranulate as well. And the phagocyte side of things just means they are a cell that eats other things. So they can engulf other cells, they can engulf bits of broken down pathogen, or if the pathogen is really small, they can engulf them as well. And they do that through a process called phagocytosis. And when they take in things through phagocytosis, they don't just let them get into themselves, they take them into the cell within membrane and they're contained, and then they put into that blob of membrane that they've contained everything, they'll put in chemicals that help destroy whatever is in there and break it down. And so they can engulf bacteria and other microorganisms or other microscopic particles and do this to them. So those are our granulocytes, which brings me to a monocyte. Now, monocytes give rise to two of our key cells here, and those are macrophages and dendritic cells. These cells are really, really important because they play a fundamental role in connecting our second line of defense to our third line of defense. Now, monocytes is where you start out with things, and monocytes are a precursor cell that circulates in the white bloodstream. So that monocytes aren't really going to give an immune response themselves, but rather they turn into either a macrophage or a dendritic cell. And once they've turned into one of those cells can then go off to do an immune job. So the monocytes can leave the bloodstream and move into tissues where they can then become macrophages or dendritic cells. So technically, if it's in the blood, it's a monocyte. Once it's migrated into tissue, it will turn into either a macrophage or a dendritic cell. Macrophage means big eater. That's what the macro means. It means big, and the phage part means the thing that devours or eats. So a macrophage is a big eater. These are really large cells that can go along like a vacuum cleaner kind of. And they are also doing phagocytosis. They're able to move along, as you can see in the time lapse there, and that's you've got the cell in the middle, it's coming along and it's moving around all those bits of probably bacteria in this case, and they're able to engulf it, take it in, and destroy it inside of them. So they come, like we said, from monocytes. Monocytes are what the cells are in the blood. Once that migrates into a tissue, it's going to turn into a macrophage, or it might become a dendritic cell. But in this case, we're talking macrophages. And so those macrophages then migrate to the site of infection. And for the most part, you'll find macrophages in pretty much every tissue of the body. They're quite prolific and they're basically everywhere. So they're really, really useful because they're able to come in, they can identify and eliminate pathogens, and they can consume them using phagocytosis. And they also help to clean up any cells that have been killed and any cell debris that might be about. They can also release molecules called cytokines, and these molecules help to stimulate an inflammatory response, which helps your body deal with that infection and to clear it up and to make sure it's all removed and gotten rid of. Macrophages, like I said before, and dendritic cells are really, really important for connecting the second line to the third line of defense. And they do this by having a special role as what we call an antigen-presenting cell. We'll dig into what that means a bit more, but you can kind of get a little bit from the name. They're a cell that is able to present an antigen. And basically, they can take in a pathogen, they can find its antigen, and then they can hold it out on a receptor molecule, presenting it to the third line of defense, going, here is the antigen, you need to recognize this. And so that's part of what they're doing between the second and third line of defense. The dendritic cells get their name from the fact they've got all these little branching bits out. The dendr bit of the word is actually relating to a Latin term that we use relating to like branches on trees. And so you can see they've got all these little branching bits sticking out. That's why they're called dendritic cells. They're kind of, I suppose, star-shaped, although it's a star with lots of little points and branches coming off it. They also come from monocytes. And like macrophages, you find them in tissues, but where they have this very different structure to them. And they behave a little bit differently. They're going to travel via lymphatic vessels to the lymph glands, and they're kind of acting a little bit like a sentinel on patrol, going around checking if everything's okay. Again, these also act as antigen-presenting cells, or APCs, and so what they can do is they can take in a pathogen, they can take the antigen from the surface, and they can hold that out to T cells in the immune system in the third line of defense. They also can do phagocytosis of pathogens. That's actually how they get the pathogen in, and they are able to get a hold of that antigen they can later present. And their presentation of those antigens, like with macrophages, is really important for actually initiating that adaptive immune response that is the third line of defense. So these guys are, like I said, super, super important in the connection between the second and third lines of defense. Now, I've mentioned phagocytes a bit already, but they are a general class of cell as well. You can have cells that are sort of phagocytic in their nature, but it's a general term for white blood cells that are capable of engulfing foreign matter through what is actually just endocytosis. And endocytosis, we've talked about previously, is a process where a cell can sort of take something inside of itself. Phagocytosis is a specific subset of that, where it does endocytosis. However, once it's inside of it, it doesn't just release it to within the cell. It actually puts, as I mentioned before, it puts chemicals into that blob of membrane it's taken in. It puts chemicals in that destroy whatever is in there, and that's how it sort of digests what it's eaten. The cells that fall in the category of being phagocytes include macrophages, neutrophils, and dendritic cells. You have some other cells that can behave a bit in this way, but they may not necessarily fall under the category of phagocyte. Now, again, these don't recognize things, your pathogens specifically. They're going through the very basic self versus non-self recognition using those pattern recognition receptors we talked about earlier. For phagocytes, some of these receptors are on their surface, some are actually inside of them, so they're able to recognize things once they've consumed them. And so these pattern recognition receptors just are able to bind to non-specific molecular patterns that they're like, hey, this is not a molecular pattern that we find on cells from this body, so you must be a foreign pathogen. And so that's what they're able to do. As I've talked about previously, this is the process of phagocytosis. It basically goes through 6 stages. There is detection and interaction, where the pathogen is taken notice of. Then it's engulfed. In engulfment, the phagosome forms, and this is a blob of membrane that surrounds the pathogen that's coming in. The phagosome is then able to fuse with a lysosome, and a lysosome is just sort of a little vesicle filled with digestive enzymes like lysozymes. That fusion occurs, those enzymes are then able to digest and break down whatever was in the phagosome. And then finally, if it wants, the cell can discharge, and basically it's like pooping out the waste and the broken down material there. So those are the 6 steps that we've got. Alright, here we have exam question 5. It says an example of a non-specific response by the immune system is A, phagocytes engulfing non-self material, B, cytotoxic T cells releasing chemicals into infected cells, C, production of memory T cells, or D, cloning of B cells to produce plasma cells. So, pause the video, pick your answer, and then you can go and check your answer with the video linked in the description below. Alright, here we have a summary of all our cells there and what they're doing, so you can read through that in your own time. Just go through and make sure you know what it is the cell does and the different characteristics of it. You will notice that they generally look a bit different, and those pictures or the drawings there generally are trying to represent what those cells actually look like. And sometimes they look similar, particularly the granulocytes can look quite similar, but you will notice that they have different shaped nucleuses in the cells, and that's actually quite a defining feature of them, not just the fact they have different actions. And that's how we're able to recognize them in a tissue sample. We're able to recognize what cells we're looking at. Okay, this brings us to exam question 6. Which of the following matches a cell correctly with its role in an immune response? A, the cell is a macrophage, and the role says stimulates inflammation by secreting interferon. B, the cell is a dendritic cell, and it says the role is presenting fragments of antigens to T helper cells. C, the cell is a mast cell, and where its role is to engulf bacteria and debris. And D, the cell is a neutrophil, and its role is to secrete antibodies. So which one correctly matches the name of the cell with the role or the job it does? You can pause the video, note down your answer, and then go check it in the video linked in the description below. So far we have looked at the cellular immune response in the second line of defense. We are now going to take a moment to look at our innate humoral immune response. And there are two key molecules that we're interested in here. Those are complement proteins and interferons. And interferons are a type of cytokine, which we'll explain when we get there. So what are complement proteins? Well, they are unsurprisingly proteins, and they complement, and in this sense we don't mean they say nice things about something, we mean that they help supplement the action of immune cells. So they're kind of supporting proteins that help immune cells do their job. The liver is actually responsible for making about 20 different complement proteins, so it plays a key role in providing these complement proteins to your body. And there are 11 major complement proteins. They have names like C1 through to C9, they have a D and a B, but you don't need to remember their names. That's just so you know why they've got those labels on them if you see them in a diagram. Where do we find complement proteins? We find them circulating around the body in your bloodstream. They're part of the plasma in there. So that's actually one of the key things in plasma. It's not just water and maybe some nutrients. It actually has things like complement proteins in it. Now, complement proteins don't do anything until they get activated. And so an antigen or a signal from an immune cell could actually be the activating thing here. And so what might happen is you have a pathogen floating about, and then molecules on the surface of that pathogen actually come into contact with the complement proteins, and that can trigger their activation. Or you could have one complement protein gets activated by somewhere else, and that's actually triggers sort of a cascade or a chain reaction where the next one gets activated, which activates the next one, which activates the next one. And so you sort of get this whole cascade of activation reactions of these complement proteins. And once this has happened, this is actually a non-reversible process. If once they get going, they're going until things are done and dealt with. So once this cascade reaction starts, it is irreversible. But what do they actually do? Generally, you can sort of condense their role into three things. And you can think of this as like PIM or PIMA. And that is phagocytosis, that's the P. Inflammation, that's the I. And then you could say just membrane attack for M, or membrane attack is MA. So PIM or PIMA. Up to you if that helps you to remember it or not. And so complement proteins help with phagocytosis of pathogens, and they do this through a process called opsonization of the antigens. They're also able to help with inflammation by attracting macrophages and neutrophils to where the infection actually is. And then finally, my favorite one, they can just attack foreign cells by rupturing their membrane and just rupturing them open. So what are they actually doing in phagocytosis? So what can happen is you have some complement proteins, in particular, there's one called C3b, and they get on the surface of the pathogen and they smother it, and they form this complex on the surface of the pathogen, and they bind to the antigens that are there. And by doing this, they just coat the pathogen in these complement proteins. The technical term for this is opsonization, and what this actually does, though, is they're like a big red flag for the immune cells, waving like a red cloth at a bull, basically saying, hey, come over here, come attack this cell. And so it signals the phagocytes, hey, this is a cell that you need to come and attack, or this is a microbe that you need to come and attack because it's a pathogen and it's not meant to be here. And so the phagocyte goes, excellent, now I know where to go, and it goes over to that pathogen and it's able to destroy it via phagocytosis. So complement proteins can interact with antigens on a pathogen, they stick to it, and they act like a big signaling flag, allowing phagocytes to know where they need to go to destroy things. So that was in phagocytosis. In inflammation, what they can do is they can come off from the surface of a pathogen, acting as little chemical signals, and they can go like, hey, I've been triggered and I found this pathogen, and they can go find another immune cell and be like, hey, I found this pathogen, you need to come over here. And they stimulate and activate those immune cells to help start off the inflammatory response. And those cells will release other chemical signals, which will attract other immune cells and other things to the site, and they get the whole thing going. This also has a specific science term, and that is chemotaxis, and that just means where cells are moving because in response to a chemical stimulus. They've detected a chemical in one area and they're moving in response to that chemical stimulus, and so that's what they're doing in inflammation. So the first one in phagocytosis, they stick to the surface and act as a big red flag for cells to come and do phagocytosis. In inflammation, they're able to be activated by having contact with the pathogen, but then they go off and they find immune cells and they get them started in responding as part of the inflammatory response. The final thing complement proteins can do is the membrane attack. This is the best one, I think, where the complement proteins, there are certain ones that can actually join together and they form a membrane attack complex, or a MAC, and they're able to punch these holes, the proper science word here is pore, in the membrane, and what that does is now there is a big fat hole in the membrane of the cell that allows all this extra fluid to rush into the cell, and the cell swells and swells and swells until it pops and it bursts, and this is called lysis. So complement proteins can actually attack cells directly. They're not just getting other things involved. They can punch these holes in the surface of the cell, allowing fluid to rush in, causing it to swell and to eventually rupture. That is the third way in which they can act. So this is sort of a summary of what they can do. You can see there, number one, they can lyse the bacteria by forming a membrane attack complex, punching that hole and causing it to rupture. You've also got opsonization there, which is where they flag things for phagocytosis. The third one is how they're activating inflammatory responses. This one also has a fourth one, which is something that we'll talk a bit more about, how they help out in the third line of defense, but you don't need to know that fourth one at the moment. All right, another exam question. This says, outline how complement proteins and natural killer cells protect the human body once a pathogen has gained entry into the internal environment. So take a moment, pause the video, write down your answer, and then go and check it against the video linked in the description box below. Now, a little bit more on complement proteins just before we move off them. This is not something you need to know in detail. However, we generally say there are three ways we can get complement proteins activated. There's the classical complement pathway, there is the alternative complement pathway, and then there's something called the lectin pathway. Now, it actually doesn't matter which pathway you go through. Once the complement proteins are activated, they do exactly the same thing. So you can sort of see we have three pathways in the diagram, and they all lead to complement activation, which then leads to the same three things that we've been talking about: recruiting inflammatory cells, causing phagocytosis of cells, and punching holes in cells to rupture them. So it doesn't matter which pathway you start with, you end up doing the exact same three things, and the result is the same. The pathogen should die. The reason we have these pathways is they occur in different stages. The alternative pathway is normally the one that gets triggered first, the lectin pathway normally gets triggered second, and the classical pathway is a bit slower and tends to be the third pathway that complement proteins get activated by. So those are there are three different ways that they can get activated. So the last thing that you've been asked to know about is called interferons. But to give you a bit of a better understanding of what interferons are and where they sit, I want you to understand the term cytokine. A cytokine is a very broad category that covers lots of different little proteins, and these proteins act as little chemical messengers in the immune system. They're very similar to hormones, but they're not considered hormones. So they're like hormones of the immune system, but don't call them hormones because they're not. There are a range of different types of cytokines, and so you can actually classify them further. You've got chemokines, which you might see abbreviated as CCL. You've got interferons, which would be IFN. You've got interleukins, which is ILS. Some interleukins also get considered lymphokines, which is just another category of molecule, and you also have tumor necrosis factors, TNF. You only need to know about interferons, but I wanted to mention the others because sometimes when you look at diagrams around the immune system, you'll notice these abbreviations for these other molecules, and I wanted you to have seen them so you know what they are, that they're molecules that you don't need to know about. Interleukins in particular tend to get mentioned as we talk about our lines of defense, and so do tumor necrosis factors. So they're handy just to sort of have in the back of your mind, oh yes, I've heard about that, it's just a type of signaling molecule, but I don't need to worry about it. So interferons have a very, very imaginative name, because you see interferons, they interfere. That's where the name comes from. cells that get infected by a virus want to warn other cells. They know that they're doomed. It's once the cell's been infected by a virus, it's kind of stuffed, it's going to die, and that cell knows it, but it might be able to sacrifice itself. Some good can come out of its death. And so what it will do is it secretes this molecule, these interferon molecules, as a way to signal a warning to other cells. Hey, I'm dying, but you don't have to. Okay, protect yourself. The virus is coming. And so they warn the other cells that aren't yet infected. And what it does in these other cells is they become more resistant to the virus. And because what they're doing is interfering with viral replication, because they interfere with the virus's ability to infect other cells, they're called interferons. Interferons generally work in two ways. One we would call autocrine, and this is where the interferon, this is sort of the cell trying to help itself a little bit. The cell secretes the interferon in the hope that it can act upon itself and trigger a reaction. The other way is paracrine, where it is released to help those nearby cells where it's able to get to. So auto means like self, and para means next to, like parallel, something next to it. Early on in infection, this is super important, and we actually have been able to isolate different types of interferon, and we do actually use interferon as a treatment for viral infections where we might not have a great range of treatments. We hopefully give someone some interferon as a way to help their cells that haven't been infected protect themselves. And so as part of the defense against viruses, interferon is super important. Unfortunately, if you get to the point where you would say the infection has developed into a disease, the interferons have really failed by that point, because they're really for that early on stage. Once it's developed into a disease, you might still have some cells that can be protected by the actions of interferon, but you obviously have a lot of very infected cells, and you need to try and deal with that infection now in other ways. Alright, time for another exam question. Interferon is a chemical that A. kills bacteria by producing holes in the bacterial wall, B. protects uninfected cells from viral attack, C. suppresses the immune response, or D. is produced by T cells. So pause the video, pop your answer down, and then go and check it with the video linked in the description box below. So how do interferons actually work? We said that they help by warning other cells, but what are they doing? How does telling a cell a virus is coming actually protect it? There are 4 things that we tend to see interferons doing in cells. The first is that when a cell receives that interferon, it triggers that new cell to start making antiviral enzymes. And so it triggers the transcription of specific genes that will code for enzymes that are able to help destroy viruses. And so that cell is now building up a stockpile of enzymes that can help protect it if that virus comes along. And so if the virus comes along and tries to infect the cell, the enzymes get activated and they can block the synthesis of the viral proteins. The next thing interferons can do is they can have an effect on plasma membrane fluidity. And so they can affect how the fluid the membrane is, generally making it less fluid, which means it's harder for viral particles or viruses to actually fuse with the membrane and get into the cell. So they sort of shore up the defenses on the outside in terms of the membrane. One of the reasons that a cell might release it, interferon, we talked about an autocrine where it wants it to act on itself, is it's going, I don't want to allow this virus to do any more damage or get away with building more of itself inside of me, so I need to die. And so that cell can sometimes release that interferon because it wants to trigger apoptosis within itself, so it dies, and so it's not able to make any more viruses. It might also be able to have that effect on nearby cells that are infected. So it can trigger apoptosis in nearby cells as well, not just itself. And finally, interferons are able to help start activating immune cells. So natural killer cells will respond to the presence of interferons, and they're going to come along and start helping out by eliminating those virus-infected cells. They're going to trigger them to undergo apoptosis. And so those are the different ways interferon is actually able to interfere in the actions of what a virus is doing. So you can see here some examples of how interferon is able to interfere with what a virus is trying to do. this brings us to exam question 9. Non-specific defenses of the immune system that act against bacteria include A. Antibodies, B. Phagocytes, C. Interferons, or D. Plasma cells. You can pause the video, pop your answer down, and check it against the video linked in the description below. Alright, slightly longer question this time for exam question 10. We've got a few different components here. Scientists have found that the milk of the tammar wallaby, 𝕃𝕚𝕚𝕟𝕠𝕟 contains high levels of peptides with antibiotic properties, as well as lysosomes, complement proteins, cytokines, and venom inhibitors. A. Name the parts of the immune system to which these peptides and the other listed chemicals belong. B. Circle one of the chemicals below found in the tammar wallaby milk and describe its role in protecting the joey against pathogens. And then scientists tested the tammar wallaby milk peptides and found them to be 10 times more effective than antibiotics such as tetracycline and ampicillin, which are commonly used to fight human diseases. The scientists are keen to find a pharmaceutical company that will support further testing and development of these peptides with antibiotic properties. Suggest what the scientists would hope to achieve as a result of further testing of these peptides with antibiotic properties. So you've got 3 questions there to answer. Pause the video, write your responses down, and then go check them against the answers linked in the video linked in the description box below. Alright, so now we've talked about our cells and our chemicals involved in our second line of defense. I want to spend a little bit of time talking about inflammation. There's a couple of other things I want to highlight though before we get there, and those are a couple of other protective processes our bodies have when dealing with damage or wounds or infection. And that is how our bodies deal with blood loss and preventing blood loss, and also fever. And so these aren't in the study design. I'm only including them as an overview because I think they're quite important to have a bit of an understanding of, particularly fever, because it's quite connected to inflammation, even though it's not technically a example of inflammation when you have an infection at a localized site. Fever is more a systemic, a whole body response to an infection of some sort. And often wounds result in infection, so how does our body stop us from losing blood as well? That's why I want to include these other 2 points. So really quickly, if you have a wound and blood is getting out, there are 4 steps that your body goes through to help prevent blood loss. The first is vasoconstriction. This is where the blood vessels contract and get smaller, so less blood is going past that area where it's leaking out of the body from. This happens in response to the nerves in the area and other molecules that get released as the cells in that space get damaged. You also have platelets starting to respond. The change in pressure as the blood is flowing through there causes the platelets to get triggered and to get activated and to start clumping together. And that is actually our second step, platelet activation, where those platelets, as they get triggered by that change in pressure as the blood is flowing through the area, they start to become sticky, they get a little bit more fragile, they start sticking together, and they start forming sort of a plug or a clot in that area. The blood is then moving into stage 3, which is clotting, where the blood is coagulating, forming these clots. You are hoping that it's not forming a clot in the middle of the blood vessel, but rather it will form that clot over the edge, over the opening where the blood is leaking out. There is a really complex series of reactions that happen here that are triggered by the damage to the cells and the release of those platelets. But there are fibers that get involved that help form this sort of network that the platelets stick to, and it forms this lovely big clot that starts to act as a plug in the hole. Once that happens, you generally don't have blood leaking out anymore on the surface. That looks like a scab. That's what that is. It is a clotted bit of blood that is closing off the hole. Once that's happened, the tissue around the wound is actually able to start repairing, and you have this new tissue that grows to help permanently heal that wound. So that is how the body deals with a wound and making sure you don't continue to lose blood. Moving on from there, when you get an infection often, or other things where you get sick, you might end up with a fever. And this is a whole body reaction. So it's a little bit different to inflammation because we tend to talk about inflammation as localized to where the infection is. But your body temperature can be caused to increase because in your hypothalamus, which is a bit like the thermostat of the body. It's a region of the brain. The set point of your temperature can get reset or put up to a higher level. And so your body goes, oh, I'm a bit cold. I need to get warmer because I actually should be, at 38 degrees or something like that. And this is somewhat helpful because there are things that get killed off in the body by having a fever. So it's actually a little bit protective in a sense. It's trying to eliminate something from the body. So if you have a very widespread infection, it might be a response there. And we find that this can be caused by bacterial toxins called pyrogens, like pyro like fire, like pyromaniac or something like that. Those are able to act directly on the brain, causing this resetting in the hypothalamus. But also your cells can release cytokines that then actually get into the brain and cause the same effect. So it's actually something, not only can that bacterial cells can release chemicals that do this, but your cells can do this because it is a response that your body might actually want to have happen. And so why is fever useful? Well, it can actually help a localized inflammatory response, and it helps those enzymes that need to work and clearing out the infection. It can actually create a warmer environment where those enzymes might work better. It also generally starts to create a hostile environment for things like bacteria. And bacteria, if they're meant to do really well at 37 degrees, like your body temperature, and suddenly it's 38, 39, and that's not an ideal temperature, and that little change in temperature can be enough to really inhibit the growth rate of bacteria and even kill some of them off. So fever can actually be really, really useful. However, if it gets too high, it can become lethal to you, and that can be a real concern with fevers, that there is a benefit at times to them, but they need to be carefully monitored because they can easily get out of control and become lethal to the individual. And it's only a few degrees in temperature increase that it can become lethal at. So I just want to touch on blood loss and fever because they are connected to, obviously often infection comes through wound opening, so I want to talk about how it deals with the other side of getting a wound. And then I want to talk, like I said, about fever because it is connected to the inflammatory response. It is just a separate thing, though, to it. It is a helpful way our body can deal with infections. So this brings us to inflammation. Inflammation is something you've probably experienced either on a very small scale or a much bigger scale. It's just a non-specific immune response at an infection. And it has 5 key symptoms. I've given you the normal English terms for those symptoms there, but I've also in brackets put the Latin names for them in case you were interested. The first symptom is a loss of function. The area that's infected that tissue can't do what it's meant to do anymore. The second sign is redness. The third is swelling. The fourth is heat. And the fifth is unfortunately pain. And the reason we have these responses is due to a range of chemical messengers and cells getting involved and causing changes in the tissue that lead to these symptoms. So what inflammation is there is an infection in the area, and this is the response to that infection. It's going to occur because your smaller arterioles, which are, they're not quite small as capillaries, but they're very small blood vessels, those blood vessels will dilate. That means there's an increased blood supply to that area. And that's going to cause a number of the symptoms that we see with inflammation. Some of the key cells that get involved in key signaling molecules are your mast cells, the platelets, basophils, phagocytes, and histamine. Generally speaking, these guys all work together to help trigger this inflammatory response. And so what they might do is, particularly things like histamine, can help stimulate vasodilation and help make blood vessels more permeable. So the arterioles get bigger and stuff can move out from the arterioles into the surrounding tissue. And that could be really useful. Things like cells that are going to help, like phagocytes, will go help and clean up some pathogens in the infected area. And so that's why you have these things being released. Here I've tried to give you a little bit of a breakdown of why these symptoms occur and how the symptoms are actually connected to the cells and the molecules that get released. So there you can see redness. You've got to think blood, generally speaking, is red. And so when you have more blood in an area, particularly near the surface of the skin, that area starts to look more red. And so as the blood vessels dilate through vasodilation, we see more blood in the area. More red blood leads to a redder color to the tissue in that area. The pain that can be experienced from inflammation can actually be due to several factors. It might be painful because there is a systemic response like fever occurring. There are molecules that can stimulate nerve endings in the area that cause this pain, like bradykinin, and histamine can actually stimulate nerve endings and make you feel pain, which is not nice. But also, if there's any swelling in the area, that can put pressure on pain receptors, again making things feel painful. And I'm sure you've probably felt a time you've had something swollen, even a pimple sometimes, that's sort of infection where sometimes that swelling, you can feel it and it kind of hurts because it's swollen and it feels like there's more stuff in that space than there should be. And that's swelling is putting pressure on those pain receptors, and that's why you can feel it from even just a pimple. Heat as a symptom of inflammation occurs because blood is warm. Your core body temperature is 37 degrees, but generally speaking, your extremities are much cooler because they're further away from the core of your body. So if you send more of this lovely warm blood to these areas, suddenly you've got more of this warm substance, and so the whole area starts to increase in temperature. And you can actually feel this if you've got an area you think is infected, aside from probably being able to see it because it's a bit red or it might hurt a bit. If you put your hand over it, you'll probably notice a bit of a temperature difference between the surrounding tissue and the tissue underneath where that infection is. And so that increase in blood flow to that area is the reason it gets warmer. And swelling is also directly tied to vasodilation, as I have the blood come to the area and have lots more blood there, and I also increase the permeability of those blood cells, and more stuff can move out of the blood cells, like more fluid can move out of into the surrounding tissues, other cells can move there, other chemicals can move there. All of that starts to cause that surrounding tissue to swell as it gets all this new fluid in it. Sometimes this swelling is a bit protective to try and create sort of this protective stuff surrounding where the infection is, particularly if damage has occurred. So sometimes swelling is trying to be protective. Normally it just kind of hurts us, isn't too pleasant to have. But it is another sign of inflammation. One thing that's not a sign or a classical symptom of inflammation, but I've put here as well, is pus, because I actually think it's really important to understand what pus is. When you have your phagocytes and everything go to a site of infection and they start to clear it up, often they die and they have to be gotten rid of. The body needs somewhere to get rid of it. And so when you end up with all these dead white blood cells and other cell debris, that is what pus is. And that's why your body tends to want to get rid of it. And a really infected wound will ooze pus because it's had a big infection. You've had lots of white blood cells come and try to kill it. They've died as they're trying to deal with this infection, and the body's trying to get rid of it. And you've got all this disgusting pus oozing out. I will also add the one symptom here that is not mentioned that is a classic inflammation symptom is loss of function, and that is something that's generally due to the tissue in that area doesn't work. It will depend very much on what type of infection you're talking about, but the cells can't do what they normally do, or swelling has been so much that maybe the area is not able to move as it would normally move. So loss of function is sort of a bit more removed, and it tends to be due to one of the other symptoms there, or just simple damage to the cells. Here you can see our different symptoms and some of the reasons they're happening. So it's just a diagram going through what I was talking about just before. So what are the actual stages in inflammation? Generally, you can break it down to 3 stages. There's a vascular stage, a cellular stage, and then a resolution stage. Within vascular and cellular, there's a little bit more information beneath that. So in the vascular stage, there is an initiation point, and then there's vasodilation. So they're not exactly the same thing, that sort of happens one after the other. In the cellular stage, we specifically want to look at cellular migration. And then finally, your resolution stage is where everything's sort of wrapping up and being resolved, and things are healing and getting back to normal. So here I just have a quick exam question, number 11. It says an inflammation reaction involves the A, release of histamines, B, agglutination of bacteria, C, production of immunoglobulin, or D, vasoconstriction of blood vessels. So you can pause the video, write down your answer, and then go and check it against the video linked in the description box below. So let's have a look at those stages of inflammation. like I said before, the first stage is the vascular stage. In here, we have initiation, which is where chemical signals are starting to be released. And then we also have vasodilation, and which is where you have the arterioles and venules, which is a sort of vein, small vessel. An arterioles, an artery, small blood vessel. In that local capillary area, those start to dilate and they start to get bigger. And then you also see this increased permeability through those blood vessel walls. And you also see at this point, after initiation and vasodilation, you also see certain clotting agents start to get released in the exudate. That's just the surrounding fluid. And those clotting agents are there to help clot formation occur so that that infection can be isolated, and so nothing is getting out of where it's meant to be, and also nothing new is coming in. So that's part of why clotting is really important. It's helping make sure we don't continue to lose blood, but it's also helping stop new infection from coming in. So in initiation, this is just the very beginning molecular response where, for instance, we've had a splinter get into the finger. As that splinters into the skin, immune cells and any that are in the area and any damaged cells in that area, they start to release cytokines. They start to release these signaling molecules. And so they're starting to send those off to help get things started, because they're going to stimulate and activate other cells in this process. If there are mast cells in the area that get triggered, they're also going to degranulate, releasing histamine. And that histamine is going to play a big role in a number of the symptoms we see of inflammation. As this happens, the histamine in particular will start binding to specific receptors on nearby blood vessels, and it's going to kick off the whole vasodilation side of things. And so vasodilation is just dilation of blood vessels where they widen, and this means more blood can get to the area. In turn, this is why we see the area get redder. We also see swelling occur and warmth in the area because blood is lovely and warm. And the swelling's happening because not only are the blood vessels getting wider, there are gaps starting to form in the wall of the blood vessel, allowing things to move out into the surrounding tissue. So this is what we talk about, increasing the permeability of that blood vessel. And so new immune cells, as they rock up in the area, they can now move out from within the blood vessel into the surrounding tissue and to go and help and clean up any infection that's there. So that is the vasodilation side of it. So that is our first part. Then we move to our cellular stage. And this is where the cells in the immune system are really stepping up and getting into gear. And they're starting to intervene, particularly phagocytic cells. And they're migrating out, so they're starting off in the blood vessel, and then they need to move into the tissue. So they migrate into the tissues. We call this migration. And what we tend to have happen here is the immune cells escape from the capillaries. That is a funny way of just saying that they move out of the capillary into the surrounding tissue. They escape. And then you'll also have neutrophils. Remember, they're the first ones who generally rock up at an infection site. They're migrating to that infection. They're the first ones there, the first on the scene. They're moving there because they're attracted by those cytokines that have been released. And as they move there, you have other immune cells following them. And the neutrophils will also release other signaling molecules to help attract even more immune cells to that site. And then eventually you have the phagocytic cells that are able to start attacking the bacteria, or if there's another pathogen present that they're dealing with, they'll start attacking them and consuming them and eating them. So just a little bit on migration. This is what happens when you have all this blood flow comes to the area and you have heaps of immune cells. They need to get from within the blood vessel to actually where the infection is in the tissue. And so vasodilation, as that blood vessel widens, allows all these cells to get there really quickly, but it also starts to create those gaps in the blood vessel wall so that not only do they get to the area in the nearby blood vessel quickly, they can actually leave the blood vessel and get into the tissue where the infection is, and they can help start dealing with the infection there. And this is actually going to keep happening until that site has been cleared of the infection and any injury that's there has been healed and returned to normal. So it might vary in intensity of what's going on throughout that infection, but it actually continues for quite a while until things are back to normal. The two big things we have here are the different phagocytes, all those different cells that act in a phagocytic way and can consume pathogens and infected cells. Those are going to be guided by the cytokines that get released by the damaged cells, and so they know where to go because there are cytokines calling them over to where the damage actually is, and then they go and phagocytose any pathogens or infected cells that are there. The other one is the complement proteins. As they go out, they're going to be attracted to pathogens, but they actually, in turn, they're going to help the phagocytes because they're going to attach to those pathogens, and then they start flagging and waving down the phagocytes saying, hey, come over here, I found a pathogen, you need to come and destroy it. So the complement proteins also getting in on the action there. So our vascular and cellular stages look a little bit like this, where we have dilation of blood vessels and we have signaling molecules being released, and then we have the cells getting into this infected tissue so that they can heal it. So based on this, I've got a couple of exam questions, exam question 12 and 13. Question 12 says, the inflammatory response is a defense mechanism that evolved in higher organisms to protect them from infection and injury. This response, A, includes phagocyte migration to the site of injury, B, is part of the adaptive immune system, C, is specific to the type of foreign body, or D, involves the production of lymphocytes. And then question 13 says, consider the following diagram showing a bacterial infection within a human. Cells moving from the blood vessel towards the bacteria are A, natural killer cells, B, would act as phagocytes, C, cause vasodilation of the blood vessel, or D, release histamine in response to tissue damage. So you can pause the video now, write down your answers, and then go and check them against the video linked in the description box below. So far we have gone through our vascular and our cellular stages of inflammation. Then we come to our resolution stage. This is where we have gone through all the the blood vessels dilating, the cells migrating, them going to clean up the issue and all of that. And now the infection's been cleared, we need to make sure everything comes back to normal, and that's what the resolution stage is. Tissue gets repaired, normal status is returned. And so what we see when infection is present, what we see in the tissue are these pro-inflammatory cytokines. Cytokines, these signaling molecules that are going to be like, yes, do inflammation, have a dilated blood vessel, have permeable blood vessels, bring all these other cells in. And you have these signaling molecules which help promote inflammation, which is a really good thing. That's what you want. That's how your body is able to repair and fix up that get rid of the infection and fix up the surrounding area. So those cytokines are really important for making sure inflammation occurs while it needs to be occurring. And so that inflammation will continue on until the pathogen's gone. But once the pathogen's gone, we need to calm things back down, and this is our resolution stage. And it's actually pretty complex because those cytokines that are pro-inflammation may still be present, and those processes that have been occurring are still there. And so somehow we need to stop and reverse what has been going on. So instead of continuing to release those pro-inflammatory cytokines, you actually need to stop having those being released, and the blood vessels need to go back to their normal state and no longer be dilated. And this actually can require other molecules, including anti-inflammatory cytokines. So whereas during inflammation we want the pro-inflammatory cytokines, once it's finished, we need the anti-inflammatory ones to help calm everything back down and bring it back to normal. Alright, we've got the rest of our video here now.
Peptides called defensins in your skin and membranes that help keep bacteria and fungi from setting up shop around inflamed or scraped skin. Which, no matter how careful you are, you're gonna get one way or another. Maybe you shave with a dull blade, or you just brush your teeth too hard, and don't get me started about the dangers of bagel cutting. So when you've breached that first simple line of defense, it's time to call on your second line of internal innate defenses. This is where your body starts pulling strategic maneuvers like firing up a fever, releasing chemical signals, causing inflammation, or other defensive tactics that help identify and attack infectious invaders. Some of the first defensive cells on the scene are your phagocytes. Their name literally means to eat, and like Pac-Man, they indiscriminately chase down intruders and gobble them up, and they come in a few different varieties. First, you got neutrophils, which are the most abundant type of your white blood cells. They kind of self-destruct after devouring a pathogen, and in fact, you've probably seen piles of their little dead bodies, because that's what pus is made of. But the bigger, tougher phagocytes are the macrophages. They're derived from monocyte white blood cells cells that have moved out of the bloodstream to occupy tissues, and some are free types that patrol tissues looking for creepers, while others are fixed, attached to fibers in specific organs, devouring anything suspicious that passes by. So when a macrophage, in say the finger I just cut slicing a bagel, sees a new bacterium coming along, it snares it using cytoplasmic extensions, reels it in, completely engulfs it, and essentially digests it and spits the rest out. And unlike neutrophils, it can do this over and over again, like a boss. But not all your defense cells are phagocytic. You've also got cells with what is by far the awesomest name of any cell in the body, the natural killer cells. You can call them NK cells if you want to, but like, why would you do that? Anyway, these tiny assassins patrol your blood and lymph looking for abnormal cells and are unique in that they can kill your own cells if they are infected with viruses or have become cancerous. How can they tell? A normal, healthy human cell contains a special protein on its surface called MHC1, or major histocompatibility complex, but if it's infected, it stops making that protein. And if an NK cell detects a defective cell, it doesn't swallow it whole like a macrophage, it pokes it with an enzyme that triggers apoptosis, or programmed cell death, which is pretty awesome. So those are some ways your innate immune cells handle their enemies, but how do they know where to look in the first place? So let's talk strategery. So say you're in a banana factory and you slip on a banana peel and you scrape your knees. Your outer fortress has been breached and the pathogens are just flooding in like orcs through Helm's Deep. Banana factories are very dirty places. Now your body wants to contain the spread of pathogens, clean up the mess, and get healing as quickly as possible, so it cues up your inflammatory response. This is basically an internal fire alarm, only it uses chemicals instead of sirens to get the message across, and instead of smoke and fire, you sense redness, swelling, heat, and pain. For example, in the event of injury, specialized mast cells in your connective tissue send out histamine molecules, and histamine is great at calling in the cavalry. For one thing, it causes vasodilation, which creates redness and heat at the site of the injury. Now, those things might freak you out a little, but they're actually signs of healing. The increased temperature, for example, ratchets up the cell's metabolic rate so they can repair themselves faster. Meanwhile, histamines and other inflammatory chemicals also increase the permeability of blood vessels, causing nearby capillaries to release protein-rich fluids. This causes swelling, which again is actually a good thing because that leaked protein helps clot blood and form scabs, while the lymphatic system sucks up and filters that extra fluid, cleaning it up before putting it back into your bloodstream. And of course, like chum to sharks, an inflamed knee is also going to attract a bunch of local phagocytes, which find it easier to escape your now leaky capillaries, and lymphocytes that are also flowing freely, helping to destroy pathogens while also cleaning up dead cell wreckage. And don't forget, during all of this, the neutrophils have been doing their best, but they were the first wave to arrive, so by this time they're starting to die in heaps. They're triggered when the injured knee skin cells release chemicals that begin leukocytosis, the release of neutrophils from the bone marrow, where they're made, into the bloodstream. To attract the neutrophils to the damaged area, inflamed endothelial cells in the capillaries send out chemicals that act like homing devices, and when the neutrophils arrive, they cling to the capillary walls near the injury, flatten themselves out, and squeeze through the vessel walls to get to work. Your big monocytes eventually roll up to the battle and transform into hungry macrophages, replacing the first line of now dead neutrophils and basically just eating up any lingering enemies and then cleaning up the carnage. Now, all this works pretty well in most circumstances, but you may have noticed with a more major injury or an especially nasty virus or infection, sometimes your local troops get overrun. When white blood cells and macrophages run into more foreign invaders than they can handle, they let loose pyrogen chemicals that tap the hypothalamus and raise your body's thermostat, calling in a systemic fever to burn everything. The resulting temperature rise increases the metabolism of your cells so they can heal faster, and it also tells the liver and spleen to hold on to all of their iron and zinc so those things can't contribute to bacterial growth. But even then, sometimes you find yourself facing a more formidable foe. That's when you call in the specialists, your adaptive immune defenses. And to learn exactly how they save the day, you have to watch next time. But for now, you learn that your immune system's responses begin with physical barriers like skin and mucous membranes, and when they're not enough, there are your phagocytes, the neutrophils and macrophages. You also learned about natural killer cells and the inflammatory response, and watched as all of these elements saved the day when you slipped on a banana.
Alright, so this really brings us to the end of talking about the innate immune system, and in a little bit we'll move on to talking about the adaptive immune system. And I really want you to understand at this point that yes, the innate immune system is giving that initial response to a pathogen, but it then has to hand it over to the adaptive immune system, because the innate immune system doesn't provide any long-term immunity. So it's able to get going really quickly, but it will at some point hand over and actually get the adaptive immune system involved so that you can develop longer-lasting immunity to a pathogen. And like I mentioned before, those macrophages and dendritic cells are going to be really important for helping to bridge that gap between the two different types of immunity. All right, here we have our last exam question. It says, other than the recruitment of healthy T helper cells, the release of cytokines, and the migration of phagocytes to the site, state 2 changes that occur during an inflammatory response. All right, so you can pause the video, write down your answer, and then go check it against the video linked in the description box below. Okay, well this brings us to the end of the PowerPoint. If there's anything you didn't understand, you can always go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I'll see you in the next video. Bye!